right? Continuing with Mendel, um, when he did his pea plants, his experiments, and he got all his results, he knew that an organism inherits two alleles, one from each parent. Okay. So by making this deduction without knowing about chromosomes, he's the one that figured out, yep, you get one characteristic from mom and one characteristic from dad, one trait or whatever. And the two alleles, again, remember, are the, like if you're talking about the colors of flowers, the two alleles would be purple and white. If you're talking about eye color, there could be brown and blue and so on like that. That's what alleles are. Okay, so two alleles at a particular site, a locus, they could be identical. Okay, and that's a true breeding plant. Okay, so if they at the two allele sites, one from mom, one from dad, and at the site they're both for purple, that's a true breeding. Okay, that means all it's going to ever have is purple plants if you do that generation. So that was his parental generation. And again, if the two alleles are different, they're called a hybrid. If you have an allele for a purple and an allele for a white, that's a hybrid. Okay, they're different from each other. All right, and again, when they say the alleles at the locus, that means on the chromosome, in the DNA, the gene, all that stuff. It means the same stuff. Okay, so another thing he said is if the two alleles at the locus at the spot are different, and you have a dominant and you have a recessive, the dominant is always going to show. Okay, it's going to cover up the recessive. When we talked about that, they figured that out biochemistry. That's because there's enzymes that turn on the dominant and leave the recessive out. It doesn't mean it's gone forever. It's there. It's just being covered up at the time. Okay, so if you determine that its appearance, it's going to be dominant. And you're not going to notice that. And now normally you would think, well, if it was purple and there's white, when you put them together, maybe it'd be light purple. It's either one trait or the other, and if it's a dominant trait, that trait shows, and the recessive trait is basically covered up. The person still has a trait, but they're not showing it. Okay? All right? So then, again, all the first generation, the first philia generation, or the kids, all had the purple flowers because of the dominant trait. But then when you got to the next generation, the grandchildren, that's when it showed up again, and that's why he knew that those traits, it was still there, it was just being covered up. He also came up with something called the law of segregation. And what that law says is those alleles separate when you form gametes. And you probably saw that in the video if you watched that about Mendel. If you have the traits, if you have the big P and little p for the color, okay, and you have those two traits, the big P and the little p, so you have the dominant and the recessive. When you form gametes, you're going to have one gamete with the big P and one with the little. So you have more variety. If you're homozygous, if you have the same, two big P's or two little p's, all you can pass on is, that either the white color or the purple color. But if you're a hybrid, you can pass on one or the other. It's a chance. It's just like we talked about with males and females. If you're XX, you're female. If you're XY, you're male. There's a 50-50 shot. Mom can only give you X's, but dad can give you an X or Y. So depending on which one you get, that determines whether you're a male or female. Okay, so thus the egg and sperm, the gametes, are going to get two different alleles if they're a hybrid. Again, if they're a homozygous, they're going to be the same. Okay, they're going to have the same gene in both, because again, once you go through meiosis, you separate those, the traits are going to get separated. But again, if you're homozygous, they're all going to be the same anyway. Okay, the segregation of alleles, well, that's his law. He said that those traits, since you get one from mom and one from dad, they separate when you form gametes. So again, that's why that recessive one has a chance of showing up again, because even though it's being covered up, once you form the gametes, well, half those gametes are going to have that recessive trait. So if those recessive traits get together with a recessive trait from the other parent, now you're going to show that trait. Okay, so that's segregation of alleles, and that means when you have characteristics, when you have traits, when you have genes, they separate when you form gametes. Okay, and that's where the 3-to-1 ratio comes in. Okay, and actually, if you did the genotypes is what the genes say. 25% of those would be homozygous dominant, 50% would be hybrids, and 25% would be heterozygous dominant. And we'll show you that through what they call a Punnett square. Punnett square was designed by a guy, his last name was Punnett, and he's the one, it showed you how you could figure out what combinations you're going to get when you break up those alleles, when you break up when you form the gametes. Okay, And again, remember, a capital letter is a dominant trait, a lowercase is a recessive. And it's important to pick, you can pick any letters you want. You know, when you look at the chromosomes, there's not little A's and B's and whatever, but we just use them to represent. But make sure there's a difference between them. Like B is a really good one. Capital B and little b, there's a difference between them. But if you use C's, it's really hard to tell the difference between a capital C and a little c. So you want to use letters where you can see the difference between the capital and the lowercase. And again, that was something they talked about in the video. Okay, so here's our purple flowers. We have, you know, okay, and again, they talked about genotype and they talked about phenotype. Genotype is what the genes say. So this PP, capital P, would be the genotype. Being purple would be the phenotype. Okay, and here we have two little p's. That's also its genotype. Okay, all right, when you cross them, okay, you get all purple flowers. But their genotype is big P, little p, because they got a big P from this one and a little p from that one. It's the only thing they could get. So that's what the gametes would have. So the genotype for all of them would be that. Okay, now if we take these guys and cross them, it was easy when you did this one because you know there's a big P here and a little p there. Now we've got a Punnett square, and this is how a Punnett square works. You take the genotype from one parent, big P, little p. Okay, this is the genotype, and you separate them because remember, they're going to separate when you form gametes. And you do the same thing for the other parent over here, big P, little p. And then you just fill it in. So one of them is going to be big P, big P. One of them is going to be homozygous dominant. Two of them, because we got big P, little p here and big P, little p here. Now, their phenotype is purple, but their genotype is big P, little p. Okay, they have a dominant and a recessive. And then you see right here, one of them is going to get a little p and a little p. They're going to get a little p for both, and they're going to have the recessive trait. That's where you get the 3-to-1 combo. And again, that's what Mendel saw in all of his pea plants. 3-to-1, no matter what trait he was investigating. Okay, so that's where it comes from. And this right here is called a Punnett square. And again, what you do is you take your genes, and or your alleles, I guess would be a better name for them, and you just, you know, because from this plant you're going to get a big P and a little p. And since we cross these two together, we get a big P and a little p, and then you just fill in the boxes. Okay, and again, the phenotype is what it looks like. The genotype is what the genes say. So the genotype would be 1 to 2 to 1, but the phenotype would be 3 to 1 because it's purple to white. Okay, so again, some of those words I was using, if they have two identical alleles, they're called homozygous, which means homo means the same. Okay, so if they're two big P's or two little p's, they're homozygous, homozygous dominant or homozygous recessive. If they have a big P and a little p, or a big B and a little b, or whatever, if you have two different alleles, then you're heterozygous. Again, hetero means different. We talked about this in other stuff before. I think we talked about these when we were talking about matter. There's homozygous means well mixed, and heterozygous means not so well. Well, same and different. Okay, so you have different alleles for the gene, and then again, you're going to have that characteristic. So you have homozygous or heterozygous. Unlike homozygotes, heterozygous are not true breeding. They call them that. They call them hybrids. Sometimes you have to help out the process as far as pollinating, but again, okay, so because there's different effects of the dominant recessive, an organism's trait, you know, you might not know that you have a characteristic like that. There's, you know, sometimes two people with brown eyes and they have a child with blue eyes, and they're like, what the heck? Well, if you're brown eyes, but you have the trait for blue eye, and your mate is brown with the trait for blue, and you end up giving him the trait for blue eyes, that can happen. Okay, if you both have blue eyes and you can't have a child with brown eyes, you guys. Nope, then there's a problem. Just saying. Okay. All right. So when you want to distinguish between the phenotype and the genotype, again, phenotype is what they look like. So blue eyes or brown hair, those are phenotypes. Genotype is what the actual genes say. And sometimes that's hard to figure out unless you know your background. Like my blood type is O, excuse me. I know my mom was A and my dad was O. Okay, my O is a recessive. So my mom must have been A and O, because how am I O if she didn't give me an O? Okay, so you can sometimes through your, looking at your parents or your grandparents figure out your genotype, but not always. Okay, so again, with the flowers, we had the big P, that's homozygous, and big P, little p. Okay, but they look the same because anytime, that first one, that's what determines it. So if you get the big P, you're purple. There's not a difference between this one and this one. This one isn't more purple or anything. It's just once you get the dominant trait, you have that characteristic. Okay, but their genotypes are different, and you would know that for the next generation. It'd be hard to tell here, but if this one has a chance of having some white offspring, whereas this one wouldn't. Okay, so again, when you were looking at, here's our purples, and they could be big P, and this, what I was telling you about when you cross them, 1 to 2 to 1. Okay, the phenotype ratio is 3 to 1, but the genotype is 1 to 2 to 1. So it'd be 1 is homozygous dominant, 2 are heterozygous, and 1 is homozygous recessive. But if you look, these guys are all going to be purple, and this one's going to be white. So it's a different ratio for the phenotype, what they look like, and the genotype. Okay, again, they do a test cross sometimes to make sure that somebody has the dominant, because if you have the dominant phenotype, are you a dominant or are you heterozygous? So they'll do, and Mendel did this before he did his experiments. What he did was to test the genotype, he did test crosses, okay? And he wanted to make sure that the offspring never displayed any of the recessive phenotype, okay? So if you take one that's purple and you breed it with, another one, and actually you could breed it with a white one, because that they have the recessive trait. If none of the offspring are white, then that one is homozygous dominant. Okay, so they do these offspring, and again, it's to test their characteristics. Sometimes people do this when they're big time breeders of various creatures. They do these test test crosses to make sure that they're okay. Okay, so again, here, if I don't know if this one is a hybrid or not, I'm going to cross it with a white one. Okay, and if I don't get any white offspring, then I know this is a dominant. And again, remember, he did pea plants, so it wasn't like he did one cross. He did a lot of them to make sure if no white offspring show up. If it is this, okay, so let's say we take this one and it's big P, little p, and over here, mom just has little p's. So half of them have to be white, okay, by the numbers, by the Punnett square. And so if you do it this way, and if this is dominant and all the offspring are purple, then I know this one is a dominant, homozygous dominant, because no white showed up. And if you got this guy, half of them should be white. So that's one way you can do a test cross, they call it, okay? Another thing that Mendel came up with is called the law of independent assortment. And what it says is characteristics segregate, and there's no real, this is one I'd always talk about, is some people think like if you have brown hair, you have brown eyes. That's not true. Hair color and eye color are on separate chromosomes, so they separate, and it's not like the brown always goes with the brown and the blue always goes with the blonde. It doesn't happen that way. They separate independently, okay? And that's what he said. He said when we did these crosses, if he started doing crosses for more than one trait, he noticed that the offspring would vary, okay? So again, just because you get one trait doesn't mean you get another, okay? So when you just worry about one trait, it's called a monohybrid cross. Those Punnett squares with the 4 boxes, those would be that would be a monohybrid cross. You're only working at one. Okay. He also looked at 2 characteristics. Okay. Maybe it was the color of the pea pod and whether it was wrinkled or smooth. Okay. And again, he'd have to do the 2 characteristics, and then he'd look at the first generation, the kids, and then he'd do the second generation. And we'll talk more about that. But when you have 2 characteristics you're looking for, you have a dihybrid cross. And instead of 4 squares in your Punnett square, you have 16. Okay. And again, we'll look more at that next time because I'm just about out of time. So we're going to pick up on dihybrid crosses next time we do notes.